I'm Jiashu from Shenzhen, and here I introduce to you a design of a CRISPR-based detection module for coronavirus that can generate fluorescent output. The coronavirus pandemic, started in late December 2019 in Wuhan, China, has now caused in total 380 million infections and 5.7 million deaths. The reason it can have such an impact is due to its high transmissibility rate, which resulted from its efficient way of spreading through droplets, and high mutation rate, due to its existence as an RNA virus. Comparing to DNA viruses, RNA viruses are more likely to mutate, as the enzymes in them lack the ability to recognize RNA damage. Also, some substitution mutations, like C mutates to U, won't be detected in RNA viruses, as RNA originally contained both C and U. Despite coronavirus spread so rapidly, the main detection method, named RT-PCR, is very complicated and needs to be done in professional laboratories, and it usually takes about 6 hours to get the result. Other than the traditional RT-PCR detection, scientists had also developed some easier ways for detection. Some of the smart bands have sensors that can detect wearers' body temperature. The problem with this is that the temperatures of different areas of the human body vary, so it's hard to find a perfect body temperature. Also, just detecting the body temperature is not accurate enough, as other diseases and illnesses may also lead to fever symptoms. As we can see, all the current methods of detection is not convenient or accurate enough, so here we introduce a CRISPR-based wearable detection module incorporated into a normal mask. The whole detection module is separated into 4 zones: the sample collection zone, the lysis zone, the RT-RPA zone, and the detection zone. The virus will accumulate on the interior of the mask due to coughing and normal breathing, in which the sample collection zone, made of materials with strong water absorbability, collects the droplets of the user for detection. The lysis zone uses chaltropic salt and detergent to break the surface of the virus to reveal its RNA information. Then, DNA primers will stick to the unique genetic sequence of the coronavirus, named viral RNA, to make sure that the test will only detect the RNA of coronavirus. By adding multiple viral RNAs, the detection module could identify different variants of coronavirus, broadening the application. The third zone will perform RT-RPA, which has similar mechanism as RT-PCR, to reverse transcriptase the RNA information of the virus into DNA, and will magnify the DNA information to make it detectable. Instead of using RT-PCR, we decided to use RT-RPA, which can be done in 20 minutes under 42 Celsius. Comparing to RT-PCR, it is more applicable on the module because it doesn't require expensive technology and due to its low working temperature. The detection zone contains CRISPR-based Cas12a, a DNA cutting enzyme. In Cas12a, there's a guide RNA or cRNA that is complementary to the essential DNA sequence of coronavirus, or PAM. When detecting PAM, Cas12a will attach to it. Then, it will unzip PAM's DNA and match it to cRNA. After that, Cas12a will cut the target DNA sequence, while at the same time cutting the quenched fluorophore probe that is complementary to the gRNA, and fluorophore will release fluorescent output. This is based on a mechanism named Fluorescence Renaissance Energy Transfer, or FRET. When the distance between the quencher and the fluorophore is smaller than 10 nanometers, energy will transfer from the fluorophore to the quencher, so there will be no fluorescent output. However, when the quenched fluorophore probe is cut, the distance between the quencher and the fluorophore increases, and FRET will no longer be functioning, so fluorescence will be generated. Here we selected methyl red as the quencher and CY3 as the fluorophore. Notice that between the zones, there will be polyurethane with weak capillary ability, which slows down the flow of the sample and cause time delays. In conclusion, we designed a CRISPR-based detection method of SARS-CoV-2 that can be integrated onto a normal mask and is capable of generating visualized fluorescent results. Ideally, the design has low cost, high reusability, high intensity of fluorescent output, and multi-channel detection ability. Also, the detection method is universal, that it can be applicated to the detection of most other viruses by changing the detection module. That's all. Thank you.